Now we are going to see about translation. This is the next process. First is transcription, formation of mRNA. Then we have seen how tRNA is formed. Now we are going to see about rRNA, the involvement of rRNA. When you see this, you will find three steps involved. First, the mRNA. So it reads the codes. The codes exist in the mRNA, and the code was read by tRNA. So after reading the code, it brings amino acids. To bring the amino acids, it has anticodes. So it brings the specific amino acids, which we have seen the gene coding. Then after that, connecting the amino acids, what is involved is rRNA. It has many enzymes which helps to connect the amino acids. Suppose one amino acid is formed and it is connected with another amino acid. This is peptide bond. To form this peptide bond, it needs lot of energy. So what happens is the ATP is already in the beginning itself. The ATP is connected with tRNA and the tRNA is charged. So it is charging of tRNA. This process is called charging of tRNA. So because it requires a lot of energy to bind to form the peptide bonds while the protein synthesis takes place. So in the beginning stage itself, ATP is connected with tRNA to supply energy. That is called the charging of tRNA. Now tRNA is charged, ready to bring the amino acids. Now the rRNA, with the help of its enzymes, makes the peptides. Then it connects with the peptide chain bonds and make polypeptide. Polypeptide is nothing but protein. This entire process is called the protein synthesis, where the mRNA brings the code, the tRNA with the help of its energy and anticodon, it brings the amino acid to the site, and rRNA with the help of enzymes connect all this, makes a long chain of protein. Protein is nothing but polypeptide, and this is the entire process of protein synthesis. Right. Now we will see it in a diagram, right? Suppose this is mRNA. This is mRNA. It has codes. Okay. Various codes. I will select some 2-3 codes very specifically. I will explain all these things. Okay. Now this is mRNA. It has a code. Suppose what will be the first code? Initiation code will be always AUG. Suppose if you just take this is A, this is U, this is G, initiation code. Then this code will be, once the mRNA comes to the cytoplasm, before the tRNA brings the amino acids, it should be read. Okay. For this, what is involved is the rRNA. The smaller code of rRNA connects here. Then the larger part of rRNA connects at the above. It is below and it is above, and it reads the code. Once it reads the code, what happens? Its parallel code is anticodon. What will be the anticode? A means U, U means A, G means C. So this is existent tRNA that brings an amino acids. Suppose this is amino acid UAC is tyrosine. Tyrosine is amino acid. which comes to the first site, and actually it is to be drawn here above, but I am just making it below. Maybe I will make the diagrams. If you make the smaller part here, bigger part here will be easy for our diagram because of the board and these things I am doing. Suppose I am not doing any change, just for our understanding, I am just made it like this. This is a smaller unit, it reads, and the bigger units has all the enzymes because all the process takes place inside the bigger unit only. Now this is anticodon, this is tRNA. It brings an amino acid. This is code. This is mRNA. This is tRNA. This is a code. This is anticode. This is anticode. Anticodon. Okay, clear? Now this is how this process takes place all along the length of the mRNA, which moves. When it moves, the next amino acid is brought and that is cut and pasted here. This is what I told you, the peptide bonds are formed and it becomes a long chain. What happens after moving to some distance? Okay, the second amino acid comes here. I will take the code and the amino acid, right? It will be like this. Suppose the tRNA comes here, which brings an amino acid, and what happens is this amino acid is cut here and this will go and join here. This is when it is moving. By this time the RNA is moving now. Like this, it is moving. Okay, the next RNA is formed. Here the tyrosine is here, the first amino acid, and it is the second amino acid based on the codes. When it is moving, suppose I am drawing here. Suppose it is the sixth place, right? So what will be that? It brings 3 codes, it brings an amino acid, and previously suppose there were different amino acids. Suppose this is TYR is here. I will give some example. Okay, any amino acid. Okay, suppose Rrna, arginine, alanine, this can be valine. Okay, some amino acids. It's a new amino acid, serine. Suppose it is serine. The chain elongates like this. Every time when it is formed, it is get cut and go and join here. This is how the ribosomes are moving and reading the codes, and the instruction is taken by the tRNA, by the anticode. It brings a specific amino acids and the peptide bond is cut, and for this a lot of enzymes are required. As I said, this rRNA contains more than 100 different enzymes. Not one or two, 100 different enzymes. That's why the role of this RNA is very important, and that cuts and that takes the other chain and connected with the newly formed chain. So the chain get elongation. See, the chain is getting elongated. Now this is called polypeptide chain. Now the protein is formed. So what you have to remember is mRNA has codes, tRNA has anticodes, which properly brings the amino acid get connected, and just connection may not help because the smaller subunit of rRNA reads the code and the larger subunit has 100 different enzymes which actually does the work of connecting the amino acids. So it brings the first amino acid and it cuts the rRNA, makes the second amino acid comes here, then it cut it and join here, cut it and join here, cut it and join, and it makes a long chain. We'll move on to the last one. Last code may be UGA. Suppose UGA means stop. The code says stop, where the tRNA doesn't bring any amino acid. So what happened? The chain is get cut. So chain is get cut, and this chain comes as a kind of a protein. This is a protein and it stops. I have drawn two dots at both the ends, and these are called UTR. These are UTR means untranslated region. Untranslated region, UTR. And this UTR, which is not bringing any amino acid, it is not translated, and which is always present at two places, at the beginning of the initiate, at the beginning of the mRNA, and at the end after the stop code, at two places. And why this UTR is there? Because they say the translation is very, very efficient, which cannot proceed further. Exact amount of protein, exact number of proteins is to be released. So it marked exactly the places where it is to be performed and it is to be cut. So it marked here and here. So beautifully it is designed. So they say this UTR, untranslated regions, are very, very essential for efficient translation. So translation is 200% perfect because of this UTR. So this is translation, formation of polypeptide chain. I think RNA world and DNA world is over. Now we will move on to one more topic. I want to take it, the genes involved in the process. Okay, the expression of genes in the process of transcription and translation. We will see in the next video, right? Bye children.